Welcome back, biologists. So, we have talked about generating an action potential, and that action potential has traveled down all the way to the end of the cell where it's going to interact and send the message to the next cell. So, this is the final step of the action potential is for the neuron to signal across the synapse to the next cell. So this area where they're not touching but still interacting is called a synapse, where these two neurons are going to send their signals. This is the presynaptic cell before the synapse. It is the one that is making the signal. This is your postsynaptic cell. It is one that is receiving the signal. Some synapses are electrical. That means that there are gap junctions between the cells that allow that electrical activity to flow straight through. That action potential just flows straight through from one cell to the next. However, most synapses are going to be chemical synapses. They're going to rely on some sort of chemical, these little red dots in the picture here, to be sent from one cell to the next. These little chemicals are called neurotransmitters. That kind of makes sense, right? They are transmitting between neurons. These are neurotransmitters. They are released by the presynaptic cell. So as this cell has had its action potential travel down the axon, it at the end will make these vesicles filled with these neurotransmitters. So when the action potential has traveled down and arrives at the synaptic terminal, it'll cause these calcium channels right here to open. So calcium is going to start coming into the synaptic terminal. That's going to cause these vesicles to fuse the membrane of the vesicle with the membrane of the cell. And when those membranes fuse, the contents of the vesicle will spew out and travel across this synaptic cleft, this space between the two cells, into the from the synaptic cleft, it can interact with these receptors on the postsynaptic membrane. When the neurotransmitters get to the postsynaptic membrane, it'll bind to these channels, and a lot of these channels will be ion channels. So the receptor binding to it will open up the channel so that potassium will flow out and sodium will flow in. So these channels are ligand-gated. So we just talked about voltage-gated channels. Now a ligand-gated channel means that when a ligand binds, in this case the ligand is a neurotransmitter, when the ligand binds, it acts like a key and opens the channel up. And then this can cause a change or a response in the postsynaptic cell, although that will depend on what the neurotransmitter was. It'll depend on the receptor, and it'll depend on the cell. Some neurotransmitters are inhibitory, and some neurotransmitters are excitatory. So an inhibitory neurotransmitter is a neurotransmitter that will interact with that postsynaptic cell, and it will bring its membrane potential further from the threshold. So it's going to make it less likely for that neuron to be activated, usually because the channel will allow chlorine to come into the neuron. So you're coming further away from the membrane, further away from threshold. Excitatory neurotransmitters typically are letting potassium and sodium through, and they will bring membrane potential closer to the threshold. So it's going to make it easier for neurons to fire or cause more excitement in that cell. So an inhibitory neurotransmitter will often result in an inhibitory postsynaptic potential. These are typically hyperpolarizations. These are kind of opposite here. Inhibitory synapses will give you a hyperpolarization. So it's getting further away from the threshold, less likely that it will fire. An excitatory postsynaptic potential, or an EPSP, will cause a depolarization, coming closer to the threshold, so it activates it. Often one excitatory postsynaptic potential is not enough to reach the threshold, and so you'll end up with a graded, localized response. But multiple EPSPs can push that cell to reach potential, to reach the threshold and create an action potential. Most neurons have many synapses on their dendrites and on their cell body, so they're able to receive many signals at the same time. When two ESPSs are produced in rapid summation, you have in rapid succession, you have a temporal summation. That's what's happening here in part B. So the first ESPS, the first excitatory signaling happens, and then before it has a chance to go back down, a second one occurs, and that pushes it up to threshold. Then you bam up to an action potential and have the response. That's temporal because it's happening over time, 2 separate times. You can also have two ESPSs coming almost the same time from 2 different locations. So this is spatial summation because they're coming from different spaces. And when they come here, they are both at the same time, and that'll push it right up to threshold. You reach your, you create an action potential and have your signaling. However, if the signals are all by themselves, here's one signal, it makes a little bit of a change in the potential, but it doesn't reach that threshold, so it just goes back down. Then later, another signal, but it doesn't reach the threshold on its own, so it goes back down. You're getting a little bit of a graded response, but not full turning on the whole action potential. So this combination of EPSPs, whether it's as a temporal summation or a spatial summation, can both result in reaching that threshold and having the action potential. Here you're summing an EPSP and an IPSP. So here's the E, here's the I separately. It's raising or lowering. When you put them together, more or less just cancels each other out. So inhibitory and excitatory together kind of tells the cell, never mind. Then after that neurotransmitter has been received, that signal doesn't want to just sit there forever. If that signal somehow is going to have to be terminated or ended. There's 2 different ways that this can happen. So let's look at acetylcholine. This is one example of a neurotransmitter. Acetylcholine will open. It acts as a ligand to open those chemically gated sodium channels on the postsynaptic cell. So sodium comes in and you get a potential. On other cells, it can stimulate or inhibit cells depending on what receptor is there. There is also a molecule called acetylcholinesterase. So you can see acetylcholine is here, but then it ends with this esterase. And that ending ase tells you it's an enzyme about to break down something. So acetylcholinesterase breaks down acetylcholine. So when you have acetylcholine there, it just gets chopped apart. And those chopped up pieces can't open the gated channels, so you end the signal. That's what is happening in this top picture here. This neurotransmitter, you can imagine it's acetylcholine, is being released. And here it's being cleaved, it's being broken down. And these fragments cannot interact with the receptors, so the transmission is terminated. Another way to terminate the transmission is to reuptake the neurotransmitter, which is what's happening in this image. So the transmitters were released, and then they are taken right back up, right back into vesicles until the next signal is ready to come. Acetylcholine is the most common neurotransmitters in vertebrates and invertebrates. It is involved in muscle stimulation, in memory formation, and learning. So right now, if you're actively participating and paying attention to this video, you're not just kind of listening in the background, but you're actively engaged in what you're doing. Acetylcholine is helping your body to create those memories in your brain. Acetylcholine is also involved in causing muscle contraction, and so it can often be associated with illnesses of your muscles contracting. However, it's not the only neurotransmitter we have. There's something like over 100 neurotransmitters in your body, and so they're grouped into these different groups. The next group are amino acid neurotransmitters. So these are pretty much just amino acids. So glutamate, GABA, and glycine are amino acid transmitters. They're active in both the CNS and the PNS. Do you remember what those abbreviations mean? The central nervous system, your brain and spine, and the peripheral nervous system, which is the nerves in the rest of your body. Biogenic amines are active in both the CNS and the PNS, and these are ones you've probably heard of before. These are fairly commonly described neurotransmitters: epinephrine, norepinephrine, dopamine, and serotonin. Neuropeptides are short chains of amino acids, so short little peptide groups. This includes substance P and endorphins. You've probably heard of endorphins before. These are both involved in pain perception. And then gases would be nitric oxide and carbon monoxide. These gases can be toxic when they work on other parts of your body at certain concentrations, but used as a neurotransmitter, they regulate local regions in your peripheral nervous system. Okay, this video is one I recommend that you watch, Crash Course the Nervous System Part 3. I'm also going to load another video for us to watch right here.
The dendrites and cell body of a neuron may receive thousands of synaptic connections from other neurons. When these neurons are activated, they release neurotransmitters that open ligand-gated ion channels in the membrane of the receiving neuron. As ions flow through these channels, the membrane potential of the receiving neuron changes at the point of stimulus and passively spreads along the membrane. Some of these synapses, shown here in green, are considered excitatory because they make it more likely that the receiving neuron will fire an action potential. Excitatory signals depolarize the membrane, making the membrane potential less negative. This change is called an excitatory postsynaptic potential. Inhibitory signals, shown here coming from the red connections, hyperpolarize the membrane, making the membrane potential more negative and making the neuron less likely to fire an action potential. This change in the membrane potential is called an inhibitory postsynaptic potential. If the neuron receives enough excitatory stimulation, the membrane potential will depolarize sufficiently for the receiving neuron to fire an action potential, beginning in a region called the axon hillock. The changes in membrane potential at the axon hillock can be measured with a voltmeter. The voltmeter measures the difference between a recording electrode inside the cell and a reference electrode outside the cell. This value is normally about -70 millivolts, which is the resting potential of the neuron. If enough excitatory signals arrive close enough together in time, they may depolarize the membrane above a certain voltage, called the threshold potential. At this point, an action potential is generated. The action potential propagates the signal along the entire length of the axon.
And here's one final little clip about how our synapses are working.
Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
Okay, so that is it for this lecture. As a little introduction to our next chapter, we're going to be talking about the brain a lot. From your homework, I hope you remember, or you can recognize, have some idea what's going on here. Who is this guy? What happened to him? What did we learn about the brain from his experience? One of my favorite stories for how absolutely miraculous, I guess you could say, amazing his story is. All right, I'll see you later. Let me know if you have any questions. I'm open for office hours, and you can email me anytime. I'll see you later. Bye.